Hello, everyone. Welcome to today's live broadcast, Extracellular Vesicle Detection and Analysis via Flow Cytometry. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Beckman Coulter Life Sciences. Beckman Coulter develops, manufactures, and markets products that simplify, automate, and innovate complex biomedical testing. More than 275,000 Beckman Coulter systems operate in both diagnostics and life sciences laboratories on 7 continents. For more than 80 years, Beckman Coulter's products have been making a difference in people's lives by improving the productivity of medical professionals and scientists, supplying critical information for improving patient health, and delivering trusted solutions for research and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speakers, Vasilis, Toxavidis, and John Tigges. Vasilis Toxavidis obtained a degree in podiatric medicine from the University of Wales. Vasilis' first position was at South County Podiatric Associates in the United States. From there, he transitioned to academic research at Roger Williams' hospital, a Brown University teaching hospital, in the pathology department. In 2002, Vasilis joined a growing Beth Israel Deaconess Medical Center flow cytometry core in Boston. There, his research interests grew to stem cell biology, where he became a crucial member of the Harvard Stem Cell Institute. The BIDMC HSCI current research interests revolve around small particle detection and cutting-edge technologies. John Tigges obtained a degree in the biological sciences from Rhode Island College. John obtained certification as an immunology specialist from the American Society of Clinical Pathologists. From there, he was recruited to academic research at Roger Williams' hospital in the pathology and clinical immunology department. In 2001, John became manager of the BIDMC flow cytometry core in Boston. There, his research interests grew to stem cell biology, landing him as a crucial member of the Harvard Stem Cell Institute. I will now turn it over to Vasilis Toxavidis for his presentation.
Good morning, everyone. Thank you for joining us. Today we are going to speak about extracellular vesicle detection and analysis by flow cytometry. All the work that we did is for research use only, and I am going to start by setting the stage for John to dive into more in-depth information in regards to extravesical flow cytometric analysis by providing an overview of what extracellular vesicles are and how we get to set our instrumentation to be able to detect those particles. So extracellular vesicles are tiny vesicles between 30 and 1,000 nanometers in diameter, and they are released to the extracellular environment from a variety of different cells. The type of cells that we have, it can be tumor cells, dendritic cells, lymphoid, red blood cells, cardiomyocytes, eosinophils, and cells from different tissues and organs. Recently, we published some work that we did working with red blood cells, looking at the diurnal variations. And we also have done extensive work with cardiomyocytes, looking at microRNA 30D and how this is a predictor of arrhythmias in cardiac resynchronization therapy. The release of EVs has been reported in the pathologies of cancer, neurological diseases, hematological diseases, cardiovascular disease, autoimmune, rheumatologic disease, viral infections, such as malaria. Traditional forward scatter versus side scatter measurement. The threshold is set to eliminate events measuring less than 1 micron. Hence, all extracellular vesicles are considered cellular debris. Small particle detection requires lower threshold capacities. The instrumentations that we use apparently were designed to look at whole blood, and hence we were focusing into lymphocytes, monocytes, granulocytes, and the red cells and debris we kind of tend to ignore in the past. So the conventional analysis of microparticles, EVs were regarded just as a simple cellular debris or fragments or part of apoptotic bodies. And the instrumentation was designed for whole blood cell analysis. Platelets were the smallest considered structure that we were able to see with a flow cytometer at the time. The way it was optimized. So the range was between 1 and 10 microns. And the forward scatter was standardized for greater than 1 micron using a photodiode and the standard collection angle of 9 degrees, which with the instrumentation that we are utilizing nowadays, we have more enhanced optics, which John is going to talk about it later on a little bit in more depth, and a different way using photomultipliers, PMTs, to collect the light with a wider angle. Also, the side scatter added a dynamic range, but the side scatter was also limited. Now we have the ability to threshold from different laser lines for side scatter and kind of enhance this dynamic range a little bit. Manufacturer threshold was set to exclude less than 1 micron in size. And adjustments allowed for about greater or equal to 500 nanometers. So that was pretty much the limit that we were used to looking at extracellular vesicles or small particles on conventional flow cytometer. Specific cytometer standards and parameters for measuring EVs were not established. This is still an evolving field. Every day we learn new things. Collaborating and sharing knowledge in this field is very important, so we can get some kind of optimization and standardization the techniques that we use, so they can be uniform as we measure those extracellular vesicles. This is a quick traditional forward scatter detection of particles. Forward scatter is a parameter measuring the light scatter less than 10 degrees as a cell passes through the laser beam. And the forward scatter measurement is related to cell size, relevant cell size. So a typical extracellular vesicle analysis with a photodiode would limit us down to 500 nanometers. Dynamic range of the original flow cytometry instrumentation, the threshold was set on the 488 size scatter and at the lowest possible setting. Depending on the platform that you use or was available at the time, you could maybe trigger a forward scatter and or side scatter and fluorescence and try to lower your threshold and try to really push the limits, try to identify those EVs. So, trying to advance the science, and we try to look carefully on the details. So we need to find a platform so we can set a range to be able to standardize and be able to detect those small vesicles. So what we did is we started by looking at dragon green beads. Standardization is very essential to achieving consistent results. And the program should be comprehensive, encompassing reagents, protocols, instrument configuration, qualitative analysis, fluorescence intensity units. So what we decided to do is to start with the dragon green beads. Dragon green beads, which we obtained from bank laboratories, they're really good for what we want to see. We want to set a range that we may be able to detect those extracellular vesicles. And that range would be, the beads that we used were between 192 nanometers and 780 nanometers in size. The reason why we use those is because the dragon green beads are an excellent surrogate for fluorescence. By using a 530-30 filter with or 525 bandpass, and the different sizes that it gives us, we're able to set the fluorescence and maybe backgate and double check the sizes in order to optimize for small particle detection. So nanobeads, they are served as a reference point for a particle detection. Sorry, I just clicked the back button. Of course, different sizes of beads that we use and different materials have different refractive indices, so it's very relative. So you have to be very cognizant when you use your beads that beads of the same size but different material, they will scatter in different areas of your plots. Now, we run the mixed dragon beads on our NanoView. We have the MoFlo XDP with our Propel Labs NanoView in it, and our MoFlo Astrios EQ. Here you can see the noise, and you can see the beads between 192, 520, and 780 nanometers. And the trigger on the NanoView was a 488 size scatter. Or the forward scatter does have a PMT alongside with the NanoView module. And the Astrios EQ was a 488 size scatter trigger on the plot that you see with the P1 mask that was provided by the company. So how do we initially try to optimize our instrumentation? So we get the dragon green beads, we sonicate them in order to eliminate any clumps, any beads sticking together. And then we take the 192s, 520s, and 780 nanometer beads, and we dilute them in PPS and 0.1% twin 20 solution with a final concentration of 1.29 times 10 to the 7th beads per microliter. 780 nanometer beads, we set the voltages on the CytoFLEX, on the Violet size scatter and size scatter properties to maximize signal to noise ratio and resolution. And we adjust the FSC voltage to maximize our fluorescence signal. The largest size is chosen first to ease of particle identification and provide instrument ability to analyze less than 1 micron. Then we proceed to differentiate with the 192 nanometer beads. So we need to differentiate between the noise and the particle. We adjust the threshold and make adjustments to decrease the influence of noise. We adjust the gain. And then finally, well, one more step, we go and run the 520 nanometer beads to verify the separation between the 190 nanometer beads and the 780 nanometer beads. So we can actually visualize the dynamic range of the instrument. And the final step is to mix all the beads together and run them through and verify the ability to separate and distinguish multiple populations. We want to make sure that we're propagating a maximum separation of bead populations for determining the relative size distribution of the matrix that we have. So the initial instrument optimization, we use the gain and threshold settings, which can be adjusted to maximize performance. However, it's strongly suggested that single bead populations be acquired again for quality assurance purposes. And the instrument has been optimized and templates and settings are saved for future reference for cellular experiments. The optimized settings is the Violet size scatter was set to a log area parameter. It was plotted against the blue 488 size scatter, again in a log area. And the blue size scatter setting was at 350 units on the gain. The FSC setting was at 370 units on the gain. And the Violet size scatter was placed at 22 units gain and a threshold of 2,000. So here are mixed beads. On to the right, you see the noise. Actually, that gate should have been open a little bit more to the right. I don't know why we put it there. And then we run the beads. So we set our gate based to the instrument noise. And then we run the beads and we see the separation between noise, 192 nanometers, 520, and 780. Now, the nice thing with those band beads is we're just looking at the scatter. There's no reference to fluorescence right now. But as John will show further down, we can, and actually I think my next slide will be referring to that as well. We can backgate from fluorescence into the scatter and double check about the populations of interest. And here's the fluorescence verification step. We have the 780s, 520s, 192s, and the noise. Please do note that the gate that you see here to the left on the 192s, 520s, and 780, these are ungated events. It's not backgated based on fluorescence. For us, it's very important to be able to, if you have a sample with fluorescence and a sample without fluorescence, it's very important to be able to distinguish the different size populations that you have. And we have been fortunate enough to have the instrumentation to do so. So, 405 nanometer wavelength versus 488 nanometers of wavelength. And it's all about the me. We have the me scatter, the Rayleigh scattering, and so the me theory predicts that scattering cross-section of the particle and thus its scattering intensity. It is dependent on the wavelength of light, the angle of collection, the shape, and the relative index of the particle. All other factors being equal, using a sorted illumination wavelength will result in an increase in scattering cross-section and thus more scattered light.
And we'll talk a little bit about the differences that we have between those two and why we're using those. So we're very interested about the me-scattering of the particles. And really quick, without getting into too much detail in regards to the physics, the me theory predicts that scattering cross-section of the particle and thus its scattering intensity, which is quite important. It is dependent on the wavelength of light and the angle of collections and the size, shape, and refractive index of the particle. So all other factors being equal, using a shorter illumination wavelength would result with an increase in scattering cross-section and thus more scattering light, which is good. We want those photons so we can see those little particles. So we decided to do a little quick and dirty comparison between the Violet size scatter versus the size scatter from the 488 MFI. And as you can see, we have the size scatter gain at 350 and the Violet size scatter gain at 22. And obviously the MFI between the different types of beads, we run 192 nanometer polystyrenes, 520 polystyrene, 780 polystyrene, and also we run latex beads, 200, 300, and 500 nanometers. And as you can see, the me fluorescence intensity on the Violet size scatter with a gain of 22, it was a lot better. there was a little bit of, which is kind of interesting, there's a little drop-off on the 520 polystyrenes and 300 nanometers latex right in the middle of the range. So I did have a question that popped up and says, wonder how to distinguish the dots are noises and not real vesicles. Well, there is no way to be able to distinguish. You are going to have vesicles in the noise region. You are going to lose some particles, vesicles, into the noise region. Unfortunately, we don't have the technology or the capability to be able to tease them out. What we have done, we have calculated the smallest size particle by using a gradient differential of predetermined particles running through our instruments, and we have determined that 30 nanometers is the lowest that you can go looking at a flow cytometer with enhanced optics. So, to answer the question, there's no way you are not going to see every single particle. That's where we are right now. Maybe in 6 months, a year, in 10 years, that will change. And at this point, I just want to go ahead and give the stage over to John Tigges, and he's going to dive in with more detail into the instrument design and give you a little bit of the science that we have done and try to explain a little more about small particle detection in our instrumentation. John?
Thank you, Vasilis. There's another question. I'm just going to look at it, and maybe we'll get to it in a second. So, actually, I'll answer that question while I talk, and hopefully it will answer. If not, please feel free to follow up. Also, hopefully we don't have any more technical issues. I hope it's coming through loud and clear at this point. Thanks again, Vasilis, for setting that kind of up for us. What we looked at here, and what this is just reflecting is, we did look at the violet side scatter using the small CytoFLEX box. Our original studies all were revolving around the MoFlo XDP and the NanoView attachment. We'll go into how that works, how we looked at the CytoFLEX and how it worked, and finally looking also at the MoFlo Astrios EQ and its dual forward scatter PMT and how we use that to our advantage with some slight modifications, of course. So what we actually did here is said basically, let's see what the differences are. Our MoFlo Astrios EQ, we dealt with looking at a 561 nanometer wavelength. We came up with this simply because, well, I guess first we said, me theory, we're going to throw it out the window. Secondly is on the EQ, on a 7 laser system, 561 happens to be the center pinhole in the 7 pinhole configuration. So we just figured more direct light. This was on collaboration with Jennifer Jones from the NIH. And so we both looked at our instruments and came up with this being the best kind of methodology at the time. We have found pretty much that any of the lasers across the board will give similar results when things are done in a certain method. To carry on, we then looked at violet, obviously, on the CytoFLEX, and blue off of our MoFlo XDP with the NanoView. For now on, we're just going to call them EQ, NanoView, and CytoFLEX for simplicity. We also, obviously, there was differences between looking at avalanche-type photodiodes and a PMT modification. So we wanted to also put that in. So we have different laser lights, different detectors, and obviously tweaks in the optics. So what we did here is then break down how does each one work. So basically our EVs on the CytoFLEX, what we wanted to do is take these guys, trigger off the Violet because of what Vasilis talked about before with me scattering saying, hey, Violet should be better according to basically a half wavelength type principle. So 405 should allow us to see things at the 100 to 200. They use what they're calling a fiber array photodiode system. Along with this comes lower noise detection, higher quantum efficiency. So something, as answering that question, something like a CFSC staining of these extracellular vesicles, basically they'll internalize, they'll get cleaved, and they will allow for the green fluorescence to appear. Not so much as you would normally think of CFSC in the premise of looking at kind of a cellular cycle proliferation. So it does work. This should work better, as it says, the higher quantum efficiency, along with what they're deeming as the wavelength division multiplexing. Basically, this is a straight shot into the detector, no bandpass filters, and you're just fiber optically bringing the signal directly into the detector itself. We tend to like to look at things, as you'll see, more from just a scatter perspective, and there's a reason for that we'll get to. Finally, we're able to control sample speed. It has a custom kind of fluidics, and so slower speeds means longer interrogation. As you know, laser beams are much bigger than the particle size at this point. So there is worry of particles sticking together, swarming effects, things of that nature that are in publication. On the XDP, with the NanoView, so the NanoView modulation that we've used in many of our papers and just other webinars or posters or talks that we've given, what they did here is they basically took an aspheric lens, focused the blue laser into a 200 micron pinhole. This way, stray laser light has all been collimated into one type pinpoint, which hits the enhanced PNT. This reduces the background. The other way they did this is it's a modified chamber. So the chamber itself is smooth instead of having angles or squares. So it's all kind of in more of a horseshoe form where everything is rounded and smooth. This doesn't allow for light to be scattering all over the chamber.
Think of a stealth fighter.
Stealth fighter. Thank you, Vasilis. I wouldn't have thought of that. So that's a good analogy. They also took the 9 degrees that Vasilis mentioned before and upped it to an 18-degree detection angle. So this allows more light to be taken in from each particle. So by doubling that to the detector, you get greater signal. So like we said, if you don't have high sensitivity, you won't be able to see these things. In our particular case, we took the largest available blocker bar that they had, which is approximately listed here as the 6.3 degrees to 12.6. Set that at approximately 45-degree angle. Obviously, there's some leeway there. We couldn't be precise. And we used the 488 side scatter. Even though this is an enhanced forward scatter, we used the side scatter to increase the dynamic range and to make more consistency. But in studies that we have done, we have shown that the side scatter and forward scatter are giving similar results. The EQ modification gives us, what we're seeing here is the actual EQ modulation. It has 2 different forward scatter PMTs set on a beam splitter here, 60-40 beam split, and they give you different type masks, they're called. The mask allow for different splitting of the laser light. The P1 was what we used. It gave the maximum opening. We also found by taking completely out the mask, you got a similar result, as it is a closed chamber, so you're not getting apparent light from other sources. This is also where we've done some cross studies with the NIH, as they also have the same configuration as us. So with Jennifer Jones, we have done some work to show that the same type work can be done in separate locations and with similar results. This is just a little bit more on what the instrument is set up as with the 7 different unique masks. We tested them all, and like I said, P1, I guess, won on what gave us the best kind of look. The other big thing is we can trigger off of any of 6 different scatter parameters. So that's where we said 561 is what we've always been using to try to keep consistency, but we've tried everything from the 405 right up to the 640. Having all those side scatters allows us to play around and plot things differently, as you will see. We're able to plot forward versus side scatter and basically threshold out a bunch of noise without having a major effect on the forward side scatter plot. Whereas other ones, as you threshold, you will see how things get cut in. So our modifications on this is we changed the forward scatter detector so that we took out the beam splitter, so we were getting 100% of the light through to the detector. We chose the P1 mask as we tested all, didn't have time to put all that data in, and then we used the 561 for our triggering. So how do these all compare, and how do we actually see it? Well, we went to what they call PCS controls. These are normal type controls for DLS type machines, and basically we took the 100s, the 200s, 300s, 500s. We did this because we expect to get similar results to the dragon beads as we were looking at polystyrene dragon beads compared to latex PCS controls. Thinking the materials in the refractive index are similar, somewhere between the 1.45 and 1.51, we expect to get similar results, but we're taking the fluorescence out so that we can see what the machine can do on scatter properties alone with kind of out the safety net of having a fluorescence back. This is due to a lot of our groups just wanting to look at things that are not fluorescently labeled. It would be great if we could do that, but we're given what the researcher needs, and we've tried to develop a methodology to do the science and see as many of these extracellular vesicles as possible. Now, just looking at the PCS, you can see here from our EQ data, we have very good dynamic range and separation from the noise, so our noise to signal ratio is exceptional, I would say, but adequate for what we want to do. The separation between the 2s, 3s, and 5s is great. This basically sets us up on a relative type setup for later stuff. We realize biologicals compared to these type samples are not necessarily the same because of refractive index, but it kind of just gives us a goal. This is the NanoView, where you can see again signal to noise ratio is great. We have decent separation between the 3 populations. Here, though, you can see the noise because we are doing a threshold off the side scatter. You get a little bit more of what looks to be this picket fencing is called. It's just binning of the data. It doesn't really know what to do with the really low stuff, so it starts to try to slam it into the higher bins. And then finally, we can look at what we got on our CytoFLEX. And the CytoFLEX shows similar type stuff. If you remember the previous slide Vasilis showed, where we had the noise here on the bottom with the gate that was a little smaller than we should have put it. You can see that we also have great separation between our 200s, 300s, and 500s, and good signal to noise ratio. Showing that all 3 methodologies had their advantages in the long run, but showed comparable results. Here we just show that in a more data format. Just everybody likes graphs, so we did that. We just compared the CytoFLEX, the NanoView, and the EQ. I know it says % difference of mean fluorescence intensity, but it's mostly just a geo-mean placement of the populations compared to one another. So between the 2 and 300, there's a very good relationship between what the instruments could show log decade-wise. And it is similar throughout the 300 to 500, with a little less separation on the CytoFLEX in the highers, which is okay with us because most of us are interested in the lower end anyways. So it's not really a big deal. We wanted to see that. So a lot of these questions that I'm seeing popping up we'll get to hopefully at the end as time permits, but I was told we could answer as many questions as possible, so hang in there. Low-end resolution is what we're really interested in. As Vasilis said, if you get very clean, clear, siphoned populations through UV absorbance, we could see down to about a 30 to 50 nanometer. In a heterogeneous population from plasma, this is not necessarily the case, as it's a little harder to differentiate who is what, and you'll see that in the end of this talk, how it dispersed the populations are. And as Vasilis so nicely pointed out, there is going to be loss in the noise. And we've seen that, as you will see, in one of the advantages of having either the NanoView or the EQ is the ability to sort. So what we did here to try to check, as we put here, we've done numerous times the EQ and NanoView to look at less than 200 nanometers. That was our bread and butter for a long time. The CytoFLEX we wanted to look at is more as a benchtop option, a little easier to use, and for our collaborators to be able to do on their own without the expertise of, say, a shared resource laboratory in flow cytometry. So we took some 100 nanometer PCS controls, we mixed those in with some 130 nanometer spherotech nanofluorescence particles, and basically what we were looking at is, okay, can we differentiate that closely related, and what is the bottom kind of resolution of the particles? So we wanted basically to see, is it still similar to our larger sorters? So we ran the 100 nanometers, we ran the 130s, and like I said, we then mixed them together, and we'll get to the bread and butter, which everyone loves, pictures. So we'll do the pictures here in just a second. So as we can see here, this is a 100 nanometer PCS control. We can see it here in the expected area. This is compared to just running the PBS that was used, and also just the instrument background, which only gave the population the dark black on the basic left-hand side. So this is how we knew that this population landed here. We also did that as a check with the fluorescence, because we wanted to be sure and not just make claims. Here is the previous data that we did on, this is the NanoView. You can see the noise, you can see the 100 nanometers just above the noise, and then the split from the 200s. Same thing on the EQ with a little better resolution, obviously, here. Great signal to noise ratio and separation between the hundreds and 200s. This also took some more modifications, obviously a little more tweaking on our micrometers. Don't want you to think that we just simply threw them in and it was that clear. When looking at the lower end, you make modifications in order to see those typical populations. As you can see here, if we would put a 500 in, it wouldn't be on this particular plot. Finally, we then looked at the 130s and the 192s, and you could see that they don't really separate clearly unless you use the fluorescence. Here in this one area, you can see the 130s appear, as they should have slightly less fluorescence on the green channel compared to the 192s. And when you separate them out, you can clearly see where they come up on the plots, on the scatter plots, which is in that region, which is then again assured us that our hundreds, when they appeared in that region before, was actually the 100 nanometer beads. So, what do we have for observations up to this point? What have we learned? Basically, what we see is that our EQ, our NanoView, and CytoFLEX give promising results and are pretty similar across the board on looking at beads for right now. And, you know, these are latex and polystyrenes. We've also done, which the data is not shown here, using the apogee beads, and so this is from, I believe they go down from 100 up to 1,000, both fluorescent and non-fluorescent bundles. So, and those showed similar results also. But that was just recently that we received those, so we haven't had time to put them into the presentations. We like the resolution and dynamic range on all three. This is allowing for more capabilities during the analysis. Also, the CytoFLEX, though, in the NanoView, as you could see, did have slight limitations. So being able to tease out the difference between hundredths and 200ths or anything in between is not as easy as it was on the EQ. And in order to get this lower signal-to-noise, better signal-to-noise ratio, our dynamic range did suffer. So putting in an actual fluorescence does clear that up. It does help. But as I said with our collaborators, this is not always the case, and we just try to use what we have to see as much of the picture as possible. So what we then did is grab some of the biological samples. We took some plasma that had been ultracentrifuged and thrown through a gradient, and basically looked at how they appeared on the different machines. So here you can see it on the EQ. As you can see, as we spoke on the actual dispersed nature of the populations in the plasma, there's many populations going on in these extracellular vesicles. We've also tested and done the microscopy to see, okay, is there lipoproteins, are there micelles, and basically we do get a very good population of what they're calling exosomes at this point, and have verified that through AFM and cryo-EM and different, even using like a Q-nano. I'm sure we're losing, like we kind of want to keep reiterating, we're losing in the noise region, but we also wanted to put some other stuff here showing its comparison to just a 500 nanometer bead. So we added that into all our, into the samples as kind of an internal quality control. This is off the NanoView using the side scatter trigger on the blue laser. Similar kind of arcing plasma profile. And finally, looking at it on a CytoFLEX, where we use the Violet side scatter versus the 488 side scatter, and you can see it gives more of a kind of diagonal plane as you're doing side scatter versus side scatter. But all kind of similar results, similar type profiles. And so we wanted to do it a little further, so we took some samples that we had on a particular study, and we did just a simple comparison using the Violet side scatter and using the 561 side scatter on our EQ. As you can see here, the results for the different timetables that we did are very similar. So we have percentage-wise 22, 23, 55, 57, and then back to a 22, 21. Very positive data on an ongoing study. So we were very pleased with how these kind of were showing the results. Pleased with the different techniques and how they all were comparable and gave reasonable data. Okay, sorry. So as a summary of going through, so that we have as much time as possible for questions, as I saw lots of them. The interest in the characterization of submicron particles or small particle detection is very broad across all of the different fields through immunology and inflammation and cardiology. So we've worked with groups doing, cardiac disease, thrombosis, malaria from red blood cells, like Vasilis said, diurnal variations across the board, just to see if, when sampling these EVs, if the time period of day was important. And basically, we are trying to establish a methodology across different institutions and to get a machine spec'd out for doing identification and detection of extracellular vesicles. And obviously, there are many ways to do this. There is fluorescence, there is scattering properties, all that flow cytometry brings to the table, along with looking at things like a microscopy, from the complex atomic force microscopy to cryo-EM, dark field. I mentioned the Q-nano, which uses a pulse resistance methodology based off of culture principles. Dynamic light scatter, which has similar problems to flow cytometry with laser-based diagnosis of these very tiny particles. But even that's come very far with looking at zeta potentials and backscatterings and multi-collection optics. The nanosite, the NTA, so that has, I guess, been listed almost as a gold standard previously for looking at extracellular vesicles, as it gave the most insight to the actual quantification of exosomes. But I'm a flow cytometrist, so my standard is looking at these guys in a flow cytometric method. How can we do that? And so we have published just recently some techniques and methodologies. We have a couple other papers in the works with our collaborators at the NIH working on these methodologies, and I mean, there is a plethora of information now and many different ways, I guess, to get to the same type answer, and it's just an idea of what is giving the most accurate and dependable data, and what can be used more broad spectrum across different universities, institutions, and stuff. So we basically have wanted to try to optimize the light scattering principles. We've looked at different types of ways to do this, obviously. There's also groups that have looked at improving their light scattering collections, and even image cytometers that will do so, that are able to check the fluorescence and in real time, and also give a picture basically of these smaller exosomes, liposomes of and the such. So what does all this still mean, right? So we have more summary. So the other things are basically that if we enhance what we have already, if we take and pay attention to the details, that was the smiley face, I guess, is looking at the details and paying attention to all those details, that we can see particles less than 200 nanometers now. That the old paradigm of looking at just 400 to 500 nanometers is no longer. And even though the conventional flow cytometer has its limitations, fluorescence can allow you to still do things on your old instruments. We're not saying you have to throw them all out, but you also can upgrade them and do things to pinpoint laser better, focus them to the particular spots, slow down your fluidics, and also, get higher optical benches on them. And basically, we looked at 3 different things, and we're able to look at them all and see how they were similar. And the big difference, though, is obviously an analyzer shows great results, but unfortunately, it cannot sort. So we still love our sorters, the EQ especially, to do look at these, give great dynamic range, and basically be able to sort out the populations for further analysis or downstream type assays, such as looking at RNA-seq. So I believe this next one's just a reference list, things that we pulled from for this particular talk today. Please feel free to do a screen capture of this while it's up, so that you can familiarize yourself with all the differing techniques and the latest and greatest that's out there. And finally, looking at how we've gotten to what we've done. We have, as I said, many posters and papers to begin with, and different characterization methods, different ways to look at these circulating microparticles in different families, and the latest being the diurnal variations of the circulating extracellular vesicles. So I believe now Vasilis and I will head over to the question and answer portion, and hopefully be able to answer your questions. I believe...
Thank you, John. I can remind the audience how to ask you questions, and then I'll put all their questions to you. Hopefully we'll have time to answer them all.
So I want to thank you both for your informative presentation. So before we get started on the Q&A session, here is how to submit your questions. You can submit them by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll try to answer as many questions as we can. And let's see. Let me know if this is the one you'd like to answer first. Can you use CFSE treatment of cells to distinguish vesicles from noise due to fluorescence?
Yeah, I tried to touch on that, and we'll go back into it to clarify. So CFSE has found itself a place in extracellular vesicle detection. There are a few groups that have published on using this technique. It is a good way to do it. The biggest problem with any sort of fluorescence tagging is still the ability to detect the lower limits. So you're now talking about having to standardize the machine using MESF type values, and also maybe looking at things in a single photon counting method. CFSE does work, but you're still looking for an interaction with the cell. So it has to intercalate and then basically lend itself to being cleaved and responding. So even in the older days when they did the similar thing with the phosphatidylserine and using a Nexin 5, you were still limited by being able to detect negatives versus positives. So it is a valuable technique also, and by marrying the two techniques, we feel that in the future it will be able to encompass more of the population to give more accurate results.
Okay. What composition of particles was used to establish the 30 nanometer lower limit? Polystyrene, latex, something else?
Okay, these were actually lipoproteins. As stated, it was a group. It's in this was the paper that was referenced previously, the characterization of circulating microparticles associated with CD39 on ectonucleotides. So this was out of Simon Robson's group here at the Beth Israel Deaconess Medical Center. And by using the UV absorbance and then taking the lipoproteins and then labeling those with a CD39 Alexaflor 48, we were able to look at them on the scatter and then look at the peak intensity on the FTSE channel. And the peak intensities were able to look at the quantitative results. So in other words, the more that he actually sectioned off in a certain line, then the higher the fluorescence peak was. And basically they corresponded exactly. So by doing that, we were able to say, okay, you have a 30 nanometer lipoprotein that is stained with an Alexaflor 48, and we were able to see that. And then we confirmed it by the fluorescence and backdating.
In our experience, height and amplitude of scatter signal is always better than area, surface, and integral. Do you ever try to compare in your system?
Yes. So area, we used, basically, we've tried height, we've tried area. The results are very similar. Most of the time we are actually using height. In this particular one, we just went because we were looking at the 3 different instruments. So we tried to keep things as similar as possible, and the easiest thing to do was to look at the area parameter on all of them. This also due to other instrumentation such as things like the influx or the ARIA, where area is more prevalent. So we just wanted it to be more across the board, but we do use height for most of the stuff that we do. We do realize that in either one, it's a half peak CV, so it's not as...
Height is more of a true measurement, so we
Correct.
tend to use that for our cell sorting purposes. So we've tried everything, and we just, as John said, want to be across the board try to compare between different platforms.
Just to give an idea, this is not in the last year or the last 2 years. We've been playing with this technologies in different ways and forms, and working to kind of tweak the instruments with colleagues at Stanford, as I said, the NIH, in other areas, even in the Netherlands and Ireland, to over probably the last 8 years to come up with what we believe to be a methodology that at least lends itself to setting your machine for its greatest chances of seeing small particle detection.
Will the system work for EVs from biofluids, for example, serum, cerebrospinal fluid?
Yes. Yes. We have done serum. We have done CSF. It's just the groups that we work with tend to do plasma, as especially with the cardiologists. We have done both samples from the cardiac sinus and through the peripheral blood, because we wanted to. Blood works the best as we're looking for how they track through the system, as everybody is looking at the cargo that they hold and how they interact in our cellular communicators.
Please clarify that latex particles cannot measure absolute size of EVs, RI index differences, but are mainly useful as a reference material for QC and standardization purposes.
Yeah, we did mention. I think I made a reference a few times in regards to this when I opened up the talk, and absolutely those ranges that we use, those beads, they're just a range. It's not absolute numbers. You cannot you cannot measure the size of EVs based on the beads. It's just a range. It gives you an idea of the range where the particles may be. If you want to do a really quick experiment, you can run some latex beads and some silica beads of the same sizes, and you can see what kind of different scatters that they have. Again, it's just a reference. And thank you for bringing that to our attention.
Yes, because no problem. Gord, I just wanted to say that refractive index-wise, we understand what has been published is that the refractive index of an extracellular vesicle is somewhere around 1.41. And basically that's why I mentioned the apogee beads, because they have a mixture of refractive index of about 1.5 and other ones that are 1.42. So this gives you more of a generalized picture of how they appear on your instrument, and also the 1.42 is more comparable to your biologicals in the fact that they're right around the same. Now, granted, they can go as far down as they say 1.36, but we still don't exactly know how their membrane reacts to light and how the actual reflection appears. It could be like a mirror, and as we see, this does happen when you can collect the backscatter. You see there's a lot of backscatter in these type particles, which would mean that they're reflecting a lot more than we actually first thought to be true.
Okay. Could you comment on measuring EV on a CytoFLEX that doesn't have a violet laser? Would it help at all to use a fluorescence trigger in this case?
Yes. Without the violet, I would try to use a fluorescence-based, for the simple fact that we have tried just straight going forward scatter versus side scatter on these, on a CytoFLEX. The forward scatter is extremely limited. In other words, there's no dynamic range off the forward scatter. If you've done this, you can see you are very limited in that aspect. So mostly what you'd want to do is probably a fluorescence marker versus side scatter. Triggering off the fluorescence will probably give you your best bet if you're doing this. And so, I guess the best way to put it is it's a lot of trial and error. It's basically figuring out best methodology for doing it on your particular instrument, and then going back and looking at also the other technologies that are available to you to solidify that what you have is real. Particles are great, as Vasilis said, to kind of set things and understand what your limitations are or what your successes can be. Whereas biologicals have their own set of problems. They're not always spherical or rod-shaped. As we know with liposomes, they're very squishy and tend to come down through a flow cell in many different configurations. So, as I said, they have their own set of issues that you have to deal with. But we like to show the beads as how can we actually get things going? And are we at least in the right framework in order to look further down? If you can't do the beads, obviously there's no reason to go looking at things that are in the 100 to 200 nanometer region on a biological. So.
Okay, the next question is, oh, I'm sorry, were you done? Were you going to add a couple more things?
No, sorry. It's just clicking on, we were looking through the questions ourselves.
Oh, great. Okay, well, here's another one for you. And this person thanks you for your presentation, as many have before. Can we apply dragon beads on older Beckman cytometers like FSC 500?
Yes, there's actually a very good paper put out by the Marseille group, I believe. So Romaric Lacroix, or there's Philippe Poncelat, the Megamix type stuff that have done work on FC 500s. It also might be Alain Bresson. So there are publications on exactly how to look at it on an FC 500 and how the Megamix can actually help with this. So we have done it on an FC 500. We have done it on the Gallios. There are marked differences between the two. Fluorescence is probably your best bet when looking at an FC 500. The Gallios then took some stuff that they had from old Quantas, and
you can still go down to 190 nanometers on the Gallios.
And we're able to open up also expand on what they call small particle detection, or they use the W2 or enhanced wide angle. So it is feasible, but like I said, it's not optimal as we're finding out. Those groups have been the same ones that have published also from FC 500 to Gallios and even some CytoFLEX data now. So they are helping kind of mold and guide how the instrumentation should be advanced.
Do you think an isolation technique for exomes matters for FACS analysis?
Oh, yes.
Absolutely does, and this is a topic that we can talk about for hours. That's where you break or make your case in regards to EV detection. What I would recommend is go through our diurnal paper and look into our isolation technique, or feel free to email us and we'll provide a copy of the protocol that our group is using. Yeah, we'll just keep this short. It's very, very important. I don't know if you have.
Yeah, ultracentrifugation is huge. There's been years of debates on what is the best protocol for doing the ultracentrifugation and which populations actually to take. As things pellet out, is it if you take the pellets, you get a lot of different stuff at the end compared to taking the supernatant. Like we said, are you getting a lot of lipoproteins? Are you getting micelles, cell fragments? All these added type things can cause some problems. We've also found in our paper that tubes that are used are a detrimental or a great success. So using different types of tubes have shown shedding during the centrifugation process. And when you're starting to look at things this small, you can actually see the pieces of the tube, the plastic that's come off. They act like polystyrene beads. You can also see things such as the dynamics of water. We've been able to look at deionized water profiles, and any kind of contaminant that actually gets in there will show up and will give certain populations. It actually took us 6 months to figure out that the tubes were all bad, and then to test a bunch of tubes to find which ones didn't cause activation or shedding.
Why are you using the 488 laser? Does it have a better scatter?
We're not just using 488. If in the talk we mentioned we trigger with the 561, and we use the 405 on the CytoFLEX, and on our NanoView we're using the 488 because that's what we have.
That's how the system was built. 488 was basically the kind of bridge between what me-scatter dictated as how low you could see. So 488 should give you around a 200 nanometer particle size that you can see, and what most instruments were equipped with. And so when you're thinking of forward scatter, it's based in side scatter. Most people think of 488 and 488 triggering. So that's where the first, when we worked for Propel Labs to try to come up with something, they went off the 488 and just instead changed the chambers and changed the optical configuration. And now other groups are trying different things, and I mean we've even heard murmurs of UVs and stuff like that. So it all really depends. There's a lot going on. It's evolving. It's just that most instruments, whether conventional as they call them, flow cytometers, or this new wave of specialized instrumentation for exosome detection, they're all equipped with 488. So a lot of people are trying to do that.
Do you recommend plasma or serum for EV detection? And do you recommend an optic purification before FACS?
I don't. Between plasma and serum, there's not a major difference. Once again, prep becomes the biggest kind of determinant of what should be done. So if you have serum samples, great. If you have plasma, great. But it's how you prep them and how you get out your EVs. And we know between OptiPrep purification, there are papers that say that is wonderful. There are kits that can be used for doing this, and basically it's what works in that particular type scenario. We like to try to see as many as possible and get the entire picture as far as we can. So this is why we use the scatter properties and look at just those type things. Everything has its advantages and limitations. Like Vasilis said, we could do probably another 5-hour webinar just on advantages and limitations of all the different techniques, which you know, there are papers put out just by that. If you look on the International Society of Extracellular Vesicles website, each year after the meeting they put out kind of a notice of the techniques and advantages and disadvantages.
Okay. We are out of. Oh, sorry.
Yeah. So Judy, if there's one or two more, we can do that, and then we're actually out of time as we have some other things to do today. To maybe look at more exosomes.
So just let me know. Do you think you have time for more questions?
We can do one or two more, please.
I don't think there's That's it.
We're looking through actually right now.
Just that last one.
Take your time.
Okay, so quick and easy ones are, there was one about, you know, is the device suitable for cell sorting? The two cell sorting ones are the NanoView and the EQ. Also, I believe the influx, modified influxes and modified ARIAs can work. And we believe cell sorting is great, but again, limitations in really small particle, large drop at this point. And so techniques are used to basically then siphon off extra fluids. That's something else that we can talk about. And I believe Vasilis flagged a question.
I can't find it.
Yeah, we saw a couple of things. I do agree with with the fact of ongoing debate of refractive indices of EVs. Like I said, a lot of these things, you go to one of these meetings and you can spend days just trying to figure out nomenclatures and techniques and, you know, what's the refractive index, how do particles relate to biologicals. It's good in the sense that it keeps the field fresh and new and really exciting.
Well, I would like to once again thank Vasilis, Toxavidis, and John Tigges for their presentations. Do you have any final comments, Vasilis and John?
No, I would encourage everybody to take a look at any flow cytometers they may have. Try the different bead arrays such as Dragon beads or PCS or Apogee beads, megamixes, just to get an idea if your instrument is suitable for this type detection, or reach out to somebody who may have something. As a lot of different groups we've learned have these type exosomes and stuff affecting their experiments, but don't know what to do with them, or even where to start. So reach out to them, see what other projects can be put forth, and collaborate as much as possible. Use all the techniques that there are to try to better the field, and we especially want to thank some of our colleagues, Dr. Danielson, Dr. Das, Dr. Guerin, Dr. Jones at the NIH, Marty Bigos and his group at Stanford, and I guess Bill Telford also down at the NIH with his laser knowledge. So, and thank you again, Judy, for moderating for us, and everybody for joining.
Thank you.
Thank you both. I would also like to thank our sponsor, Beckman Culture Life Sciences, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through July 20th, 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.
Bye.